In this lecture, we're going to continue our examination of the action potential and the role of the sodium-potassium pump, or more appropriately termed the sodium-potassium ATPase. And this is a protein functioning as an enzyme that exists embedded within the membranes of cells, particularly embedded in the membrane of neurons and muscle cells. And the function of this ATPase is that it not only functions as an enzyme, but also functions as a transporter. And as a transporter, it will move 3 sodium ions out of the cell while moving 2 potassium ions into the cell. And in doing so, those ion movements works uphill or against the electrochemical gradient. And because the ions are being moved uphill, it necessitates the expenditure of metabolic energy in the form of ATP. And so the protein here will have binding sites not just for the ions, but also for an ATP molecule. And within skeletal muscle cells, the function of the ATPase, of the sodium-potassium ATPase, is to establish and maintain the electrical potential of the cell under resting conditions. And so this sets up the energy state, or rather the potential difference. And by ensuring that the majority of sodium ions exist outside the cell and the majority of potassium ions exist inside the cell, that provides the reason for activating the cell when it is triggered by either a ligand or a voltage. So in order to ensure that the cell remains in that electrical potential state under resting conditions, the sodium-potassium ATPase is functioning. And the second major function here, operating through the same mechanism, is to reestablish the cell towards that ionic equilibrium that existed under resting conditions following a disturbance. And the disturbance is the generation of an action potential. And so when an action potential is generated either through the opening of ligand-gated channels or voltage-gated channels, the sodium-potassium ATPase will increase its activity up to 10 times of the activity under resting conditions to ensure that those ions move back into that resting equilibrium state, and that will increase energy expenditure. The protein is structured as 2 major subunits termed alpha and beta. The alpha subunit is the larger of the 2, and it is what is referred to as the catalytic and transport unit of the enzyme. In other words, it has a binding site for ATP. ATP will bind to a small region of amino acids that will generate the chemical reaction to harness the stored chemical energy that will be transformed into heat and transformed into mechanical energy that is necessary to move the ions uphill. It also has the binding sites for said cations, as well as the binding sites for an experimental inhibitor referred to as wabane. The beta subunit is the smaller of the 2, and its role is to modulate the affinity of sodium and potassium and contributes to the normal maintenance of the overall function of the protein. This structure here, this drawing, illustrates the 2 basic components of the sodium-potassium ATPase. As you can see, the larger alpha subunit on the left, where we have the extracellular medium and portions of the protein existing on the exterior side, while the majority of particularly the larger alpha subunit exists on the cytoplasmic side. And what we see here is that we have the sequence of amino acids that are numbered according to the start amino acid, indicating by the amino group and terminating at the carboxylic acid end for the alpha subunit. And likewise, we see the amino group of the first amino acid of the beta subunit terminating at the carboxylic acid end of the beta subunit as well. The green, blue, and red amino acids indicate areas where different isoforms have different residue or amino acid substitutions, indicating that although we will be discussing the sodium potassium ATPase function generally here, this protein does exist in different isoforms depending upon the cell. So this ends the first part of our discussion on the sodium potassium ATPase.